Here we go. I'm clicking the little button and going live. And let's see how this looks. Okay. Well, hello. Oh, we're still 1 minute early. I'll just start warming up here. La, la, la. Yeah, it's pretty easy to warm up a monotone. Okay. This around, I got windows on. It takes a while for YouTube's lag to catch up. All right, but we are streaming just with this funny lag that YouTube imposes on everything. So let me just introduce myself. I'm PZ Myers, if you couldn't tell me from all these other people. This is a patron Q&A stream, but of course I also accept anything from anybody. If you've got questions, go ahead and fire them out. I have got 2 questions lined up, but also big surprise, I actually have 3 people who showed up today for this little session. So wonderful. I'm glad to see you all. So let me have each of you introduce yourselves, and we will start with Jody, since she's right next to me in my little list here.
Hi, PZ. I'm Jody Baker. I'm a physics and astronomy professor in Michigan. So I had mentioned that I thought maybe you might want to talk about going back in the fall. So I thought I'd come here and talk about it with you as well.
Yes, that's one of the questions on my list, is all of us uncomfortable faculty who are expected to go back into the classroom. What's that going to be like? What are we doing? Yeah, you asked the most disturbing question of them all.
It's very disturbing, but you know.
Okay, next up, let's hear from Jason, who is making pancakes.
Okay, now a cabinet that isn't messy. I'm Jason, I'm from British Columbia, and I'll be making pancakes. I posted a question in the later Q&A section about glowing spiders, glow-in-the-dark spiders.
Oh, yeah. And unfortunately, some spiders naturally glow in the dark. Well, they don't glow, they reflect. But I don't know of any commercial ones that are being made.
Could be a moneymaker.
I have discovered that the popularity of spiders is kind of low, so we'll see. Okay, next up, Thomas.
Howdy. Well,
Hey
I'm
there.
here in Minneapolis, Minnesota. I'm an old retired guy, but I was at the University of Minnesota for many years. Excuse me. It's not COVID. But I was in the, technically I was working in the Department of Astronomy, but physically I was helping running a computational physics, or yeah, I guess it was computational fluid dynamics lab. And we were in Walter Library, which was renovated in 2000 for $65 million, almost as much as it cost me to put a kitchen on my house. But anyway, if you've never been to Walter, it's worth going just to see the building. It is just an amazing piece of work. So anyway, but now I just do, I'm still associated with the university. I'm on a contract. I just do a little bit of consulting here and there for the, which is now the Institute for Astronomy and Astrophysics, I guess. They're in Tate Physics Hall, which also was renovated. They gutted the building. The only thing standing were the 4 walls. And it is an amazing building now. It's really, really nice. What was the other thing? And I teach science and math to the homeschooled kids in the neighborhood. And the reason I got teaching them is because their mother came and said, their teacher told them the earth is 6,000 years old. Is that right? I said, no. So anyway, I've been deprogramming them, and it's been a blast. They're middle school kids.
Uh-huh.
So, and by the way, is it Jen? Who is the woman who spoke?
Jody.
Jody. So you're in Michigan. Are you from Michigan? I'm from Michigan. I grew up in Detroit. So.
Yep, I'm in the Detroit metro area.
Oh, really? Okay. Yep. I grew up in the Northwest Side, Greenfield and Puritan, which is Greenfield and 6 Mile. But
Yeah,
anyway,
I know where that is. Not too far from there.
that's my old neighborhood.
Cool.
Anyway, I'll yield the rest of my time.
Okay. So let's just dive into these couple of questions then. And you guys, if you want to interrupt at any time and offer your opinion, that's great. That's all fine. So the question was about, as a faculty member, what am I doing this fall? And oh man, it's been an unhappy summer where I've been struggling with our administration, trying to get them to face reality. At the start of the summer, the word was, oh, no masks, no social distancing. No, oh no, we're not going to require vaccination. And so the idea was, you guys should all be going back into the classroom, and you should all be, freely interacting with the students. And over the course of the summer, it gradually got a little bit more sensible. So then at first they got rid of the dismissal of masks. So now everyone is expected to wear masks on campus, in the buildings. Don't have to wear them outside, but that makes sense. That fits with CDC requirements. They're still pretending that we can pack students in. So we have new social distancing guidelines of, you're supposed to social distance by 3 feet, 1 meter, which is kind of silly because that's what we space the students out just normally, and they will cross that boundary all the time. And now the good news is that they've said that they are going to mandate vaccines as soon as the FDA approves them. Okay, well, why not just go ahead and do it now? We have vaccination requirements to attend the university, just not about COVID. But so just add this one. We'll see. I'm hoping FDA will get off their butts and we'll get it done really, really soon. Unfortunately, what happened was that last spring, they committed me to teaching an in-person class, our introductory biology course. And this summer I wrote and I asked, can I change that? Can I go to fully online? And the administration came back and said, no, you may not. This is not permitted. That the students signed up for an in-person course, so you are going into the classroom, which doesn't make me very happy. I'd rather we were safe about all this and erred on the side of caution. So my plan is we start classes in, oh my gosh, is it next week? It's something like that. No, the week after. And I'm informing all the students in the class that the first lecture hour is going to be online. And during that lecture hour, I'm going to poll the class and ask them, do you object if we just give up on this nonsense and go fully online? My hands are kind of tied though, because if any one student says, no, I want it in person, Well, because of the way the administration thinks, I'm going to have to do it in person. So, yeah, we'll see. As it stands, those students better not expect any hugs and kisses from me.
Can I ask you a question? Can I ask you, do you have a lab component to that course?
Yes. Oh, well, not to the intro course, but I'm teaching another course in cell biology. And yeah, we have a lab component there.
I mean,
What-
I'm in kind of a similar situation that you are, where the school first was like, and everything is, fine and no masks and no social distancing. And then slowly as the summer has gone on, now we have a mask mandate. But we don't have any real social distancing mandates other than we would like you to do it. I'm having a hard time trying to figure out how the heck to do labs with.
Right. Yeah,
So-
last year, the way we did it is we did have that social distancing requirement. So the way we did it is, for instance, my genetics lab, I split all the labs in 3. So each student came for a third of the lab hour for instruction. And there I would explain to each one what you're going to do. And then we just sort of had it, you come in whenever you can to do the actual work. And that was manageable in genetics, because genetics, you're at the mercy of the fly's timing anyway.
Mhm.
So yeah, but it meant that the lab was a little weird in that the official lab hour was just, here's where I explain things to you and tell you what you're going to do, but you're not going to do them yet. You're going to come in this week and do them.
Mhm.
Yeah, this year though, we're just packing them in.
I don't understand we've been given no official reasoning on why we're just going back, and we're not at full capacity. We're at 80% capacity, but that's like 4 students per class, basically, for what for our containment. So it makes no sense, and nobody's given us any really good, this is why we're doing this. And we have a lot of people in science that are extremely concerned, as you can imagine.
Oh yeah. Yeah, what I predict is going to happen is, well, let's take the optimistic prediction is that
Yes.
Delta doesn't hit our university and then we're fine. Then we'll just get the job done.
Well, you're, I think, probably out in a little more rural place
Yes.
than I am. Delta is already screaming rampant where I am. So,
Oh yeah, well, like I said, that was the optimistic
Yeah.
prediction. Do I believe it's going to happen? No,
yeah.
no. And yeah, we are in a very rural area, but we're in a small town. The local school district has rather aggressively announced that there'll be no masking and no vaccine mandate and all this kind of stuff for the public schools. So they're going to be a source of problem. We've got, a lot of the faculty here have kids who are enrolled in those schools.
Right.
And it's not going to be pretty.
No.
Yeah. So my pessimistic prediction is Delta hits. We see rising numbers of students going into quarantine. Somebody gets really sick. And then the university shuts down like it did in spring of 2020. And they'll just send all the students home. We're going to do everything online for the rest of the semester.
Yeah, I was just reading about some of that this morning, like down in Louisiana, where they've been hit pretty hard. There was a particular church, I guess, where 6 members died in the last day, and they're all under 35.
Oh.
Yeah,
So
I mean,
it's hitting a young population very hard.
right. Yeah, in spring of 2020, we were concerned because we got all these retirement homes around here and older folks living around. And yeah, then we say, no, we better do something to protect the community. But yeah, this new variant is hitting young people of the age that are going to the university. And I don't want to deal with dead students or dead students with chronic ailments for the rest of their life. I don't want to be responsible for that. the university is so confident, though, that that's not going to happen. They may have to deal with it. Yeah, and realistically, all the rules we could impose, we can only impose on the university. And these are 18 to 22-year-olds. They're going to do stupid things.
Well, I'm a little worried because we've been told that if students aren't wearing their masks, we have to call public safety.
Oh,
And I'm worried about, that's going to cause some friction if I have to be
yes.
constantly calling public safety. So I'm hoping that, the first day I can say, look, guys, just keep the masks on. I guess we'll just take a lot of breaks.
Yes, I have my student body here it's actually a very nice group of people, because they're mostly from the region. So North Dakota, South Dakota, Minnesota, Western Minnesota in particular. And a lot of them are farm kids, and they come in and they are so polite. It's creepy in some ways. They're very polite and they're very friendly, and they try to be obliging. So, I'm kind of expecting it can go pretty smoothly. I won't need to call health and safety. I'll just say, hey, Kate, please, could you put your mask on? It's a requirement here. And they will politely put it on. That's been my experience.
My wife was saying that a friend of hers is, I guess, her brother's daughter's getting married, and that whole side of the family is unvaccinated, and she doesn't want to go to the wedding. So I found that you can buy for $160 a hazmat suit to wear to the wedding. So you can wear one of those to class and kind of peek out through the, say, okay, who's not vaccinated today?
Yeah. Yeah, I've got, in 2 weeks, I have to go to the West Coast. I'm going to my son's promotion. He's in the Army. And I don't think there'll be any problem with disobedience. Everyone
Yeah.
will
By the
put on
way,
their mask.
speaking of airlines, there was a thing on, I think it was on NPR. No, it was on the PBS news Friday or Saturday. But anyway, they were talking to a guy from United Airlines, and the president of United was talking about, I think they were going to require vaccines for all of their employees, that kind of thing. But the other thing he mentioned was the filtration system in the airplanes have
Yes.
been significantly improved.
Oh,
And he said they push the air through these modified HEPA filters that will filter out the viruses and everything else 30 times an hour. He said, so one of the safest places you can be is on an airplane, even with all those people. So
I hope. I mean,
yeah,
Delta is not making that requirement for their
for the
crews to be.
employees, right? But their
Yeah.
airplanes may have been retrofitted with these
Oh,
filters and things. It'd be interesting to know. I can look that up and figure it out. But
yeah. That's reassuring, because my
yeah,
whole family is getting on an airplane. Oh boy. Okay. Yeah. And
yeah,
we're all vaccinated. We will all wear masks, but one of the people on that plane will be a 2.5-year-old who is not vaccinated yet. So
yeah.
yeah.
Mm. Well, that's a
Yeah.
little bit of comfort knowing that, because I think we have to get on a plane in October as well, or we might just drive. We're coming to Grand Rapids, Michigan. So,
Oh,
and I don't know what those people over there think about at this particular wedding we're going to, if they're going to be vaccinated or not.
Well, it's the west side of Michigan, so,
Yeah.
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 So that means no vaccines? Is that what you're telling me?
Well, I don't want to get political.
Oh, you can get political. This, my side of Minnesota is pure Trump. It's just red state on
Yeah,
this.
that's
Okay.
Trump country over on the west side of Michigan.
And the west side of Minnesota. And
Oh
yeah,
my.
and their vaccination rates are pretty low. I think it's I think it's something like 50-60%, somewhere in that range around here.
Mm.
But of
Yeah,
course,
I guess
nobody
I'm-
wears a mask anywhere.
Oh, no.
I guess I'm fortunate to be in a Democratic district. So actually I'm in Keith Ellison's, what
Oh.
used to be Keith Ellison's district before he became a state attorney, and our state legislator representative Connie Bernardi has been a good friend of mine for even before she got into politics. So go to her parties, and I got to meet Keith several times and found out that his wife and my sister used to swim together on a swim team, and we grew up in the same neighborhood. Anyway, but he's younger than I am, so I never crossed paths with him, but he's a super nice guy. I mean, he's got a lot of integrity.
Oh, good. Yeah. No, I can't say the same of my representatives, but
Yeah. Yeah. At least you don't have like Matt Gates or somebody.
oh, right. No, we've just got these rural agricultural specialists who hate ecology and just want to plow everything up. And hey, this COVID stuff is nonsense. So yeah, that's what I have to deal with.
It's pretty aggravating.
Yeah. Yeah, I just live in a little tiny Democratic bubble within a red state county. But the university all around here, I looked up the stats and we're something like 90% Democratic in my little neighborhood. So.
Mm-hmm. It's kind of like Austin, Texas, right? They're a very Democratic bubble inside of it.
Right. Yes, but there's not enough of us to overwhelm all the Republicans surrounding us. So
yeah.
So we're dealing with it. So yeah, that's how we're coping with the pandemic. Is the university is slowly seeing reason, and we're trying to rush them along. And I think the last thing that'll convince them is just sick kids, which is disgraceful. Yeah. Maybe sue them. At least that's my threat. I've put out a notice that I said, if your kid gets really sick or dies at the University of Minnesota, just feel free to call me, have your lawyer call me, and I will testify that I sent letters to the president of the university, to the chancellor of the university. I said, this is nuts. You got to do more. And they never replied to me. I don't know why. Yeah, so I hope things are better out there in Michigan. Hope they're better in the Twin Cities. Yeah, we'll see. I'm struggling.
By the way,
go ahead.
No, I was just going to ask Jody what her field of expertise is in astronomy. I'm always interested in astronomy.
Well, I'm a variable star person, so
okay.
Yeah, AAVSO stuff, and I'm mostly a teacher though, so my focus is instruction, not research so much.
Okay. Yeah, that's kind of
really
where
cool.
I'm at. Yeah, I'm at a liberal arts university. What I get promoted on is quality of teaching and things like that. But they do want us to keep our hands in, keep doing research.
Yeah.
Yeah.
Yeah, I don't have any funding for research, but I just recently joined the NASA Community College Network, and they're giving us nice, pretty telescopes.
Ooh.
So that we can do some projects with our students. So, I kind of have to find the money to do research with students. It's not something that's given to me. So
yeah. We actually tap into this grant and aid program at the university from, Twin Cities has more money than we do. It's like right now we're in the midst of putting together a grant proposal to get a confocal microscope here on campus.
Oh, that's great.
Yeah, that would be really wonderful if we did. It would allow me to do a whole bunch of new things. So
that's great.
Yeah. So we got the telescopes turned around the other way is all. And
Well, I love all sciences. In fact, I have to say, PZ, that ever since you started your spider research, you have done a service to me because I have been frightened of spiders for so long. But I signed up for your Patreon with the spider thing on purpose
oh,
because I wanted to learn more about them. And now when I see a spider crawling across my wall, I don't scream. I look at it and go, I wonder what kind of spider that is. So you actually helped my what spider phobia. I don't know what that.
oh, good. Yeah,
Yes. Thank you.
arachnophobia. Yeah. I know, and it's kind of good for me too because before I didn't know anything about spiders. I'm a newbie at this, but like just this morning, I went into the bathroom and discovered that there was this gigantic eratogenia that was living behind the radiator that crawled out and just sort of looked at me and waved a leg at me. It was kind of nice.
So,
But
so
yeah, it
PZ,
was,
how did you get from cephalopods to spider? How did you change your interests? I mean,
well, I'm still interested in cephalopods as examples of alternative body forms and that's what really kind of interests me is these variations in the evolution of life on Earth and how animals are so diverse and they're so different and all these different forms. And there was just a period of reckoning when a couple of years ago when my zebrafish just stopped laying. They do that periodically, and it seems like every August, for instance, there may be some seasonal factor involved in this. They would just stop laying eggs, and then everything grinds to a halt in the lab. And I said, this is really annoying. I wonder if there's something different I could do. And then I started doing research, and oh man, spiders, they're everywhere.
yeah.
Yeah, I can just go. At that time, I could just walk into my garage and I could find spiders and egg sacs, and there I can start doing experiments with these. Let's do it. Let's get a colony going, so I can start doing.
I walked into my garage the other day and walked through a gigantic spider web that was the size of my head. Of course, I didn't see it, but I could feel it. I'm thinking,
Right.
oh my God, where is the spider?
Yeah,
So,
these cobweb spiders, they kind of lurk. They don't hang out in the middle of their cobweb. They don't need to because they can sense when things touch it. So they're all hovering up there in the rafters or something. And if something edible bounces off it, they come scurrying out and take care of it. Yeah, so it's been a bit of a struggle to master spider husbandry. The first year, I did not have anywhere near enough spiders to maintain it for a year, and last year was kind of marginal. So this year, my focus is I got to expand this to like 100 or some more spiders so that I can reliably get egg sacs even in January, when normally all these spiders are dead. so I've just got to keep them warm. But yeah, it's also been fun because switching topics like that, it's very invigorating. When I was in graduate school, it drove us crazy because our advisor, Chuck Kimmel, whenever he got a sabbatical, he'd go off somewhere and do something completely different from what we were doing in the lab. And then he'd come back with all these new techniques and protocols and cool things we could do. And so all of a sudden there's this upheaval where everybody in the lab is changing over and we're bringing new students in who have to master these new, it kind of drove us crazy, but now I'm realizing that's the way to keep your mind fresh and young and do exciting things. So I did it too. Thanks, Chuck. You set a good example. Okay, let's answer Jason's questions. He's asking about fluorescent spiders because you can go to the pet store now and you can get these zebrafish that had incorporated into them genes for fluorescent compounds, fluorescent proteins, and they glow all these pretty colors when you shine a black light on them and stuff like that. No, they tend not to be quite the same thing. The thing with zebrafish is they have thin skin, thin transparent skin. Their pigment cells are right there on the surface. And so they can do a pretty good job of being visible when they're glowing. My spiders know I don't know of anybody who's doing that stuff. Yeah, the other thing is that spider pigments tend to be deeper beneath the cuticle because spiders are going to molt frequently, right? They're going to shed everything. And so they have their pigment granules down deep below that. So it makes it harder to manipulate them. They're also a new model organism, relatively new. So people haven't been doing the molecular biology to insert those genes into them, which is too bad. Maybe in a few years, we'll see. But otherwise, right now, if you go out at night with a dark light, I've got a UV light somewhere around here, and I've done this a few times. You can catch their eye phosphorescence fairly easily. They'll do that. The spiders I study have a tapetum in just the dorsal pair of eyes. So if you go out late at night and you shine a light out there, you'll see, like cats have a tapetum that reflects the light and makes their eyes glow. Same with thing with these spiders, but it's just two of their eyes. So you can go out to a bush and you shine a light in there, and there's all these little glowing pairs of eyes looking back at you. It's kind of neat. But lots of spiders do that. Scorpions, of course, have fluorescent chitin. So that's one of the neat things about studying scorpions, which we do not have here in Minnesota. So yeah, I'm sure some of my Midwestern followers are happy to hear that. We don't have scorpions here. But in the desert of Utah, Arizona, places like that, one of the ways you can find spiders, you just go out at night with a black light and you get these greenish blue glowing scorpions scurrying all over. Might give you the heebie-jeebies, but yeah, because there's lots of them. But yeah, you can do that as well. So there's no need to implant genes for this in them. We have no idea why they do this. Tapetum makes sense. That's to increase the light sensitivity, the photoreceptors. But yeah, the other stuff, I don't know. Just chance, I guess.
Yeah, I had a black emperor scorpion years ago. And if you're ever looking for it, a black light was a sure way to find it.
Yep. Yeah.
Very cool.
Yeah. When I was a kid, I had a pet scorpion for a while. It died. I kept its corpse in my windowsill though, because they're so cool looking. But
I have to admit, all I had was an album from the Scorpions. That's dating me.
yes, it is. That's putting you back in my generation.
I'm just about a year younger than you.
Yeah, I could tell.
Yeah.
The beard still hasn't gone quite as gray as mine, though.
Yeah. Well, only my hairdresser knows for sure, and that's my wife. And you can recognize that particular slogan.
Yes. Yes, I can. Okay, let's go on to my next question. So I was asked about the neutrality versus selection wars. Did you experience that in your career or back when you were a student? That's kind of an interesting question because it says something about the nature of science. The press jumped on this. There was all kinds of, some of you may have seen the photos of Stephen Jay Gould's group dumping buckets of water on the head of Edward Wilson. There was all this conflict about this stuff, but that's not how science works. It really did not matter to the scientists who were doing the work. So there were no neutrality versus selection wars. I knew at that time several students who were population geneticists, hardcore math and computer science people who were doing all these analyses. They were doing morphometry. They were going out and measuring. This was when I was at Utah. They were doing desert plants and measuring dimensions and parameters of their growth and all this kind of stuff. And also grinding them up and getting molecular data and pouring it all into a computer that would then grind away for weeks doing their analyses. And they had no problem with any of this, because the way it works is, if it works, you use it. So they're doing all this stuff. They're incorporating all this neutral selectionist stuff into their models that they were doing the analysis. Partly because it was a parameter that made sense. Yeah, okay, this is how it works. We'll just do the analysis with that. And partly also because if you tried to publish your work and you sent it off, a peer reviewer would come back and say, but did you consider X, Y, and Z? So yeah, you got to consider all that stuff. So, you know, at the level of the people doing the grunt work and doing the analysis and actually doing the data collection, no worries, not a problem. Yeah, this is just a new thing to fold into our work. And it was just taken for granted. You know, if it didn't work, they wouldn't have used it. So all that stuff on Kimura and OTA and all that, there was the math. Yeah, we can put that in our computer model. We should put that into our computer models and continue the analysis. No problem at all. Now, I was not a population geneticist. I was not doing that kind of thing. I was a developmental biologist, and I hung out with developmental biology people. And we were more caught up in the so-called Evo-Devo revolution, which again was not a revolution. It was just a matter of course. Here's some observations. Can you make sense of them? How do you make sense of them? How do you incorporate that into your work? So one good example of that is this was in the 1980s, and I was doing all this work on nervous system development, and I was working on insects at that time. And Yanni Nusslein-Volhard and Eric Wieschaus came out with all this amazing stuff about fly development and fly genetics, and early genes that expressed in early developing embryos. And I happened to be working on early developing embryos myself. So this woke everyone up because they said, hey, we ought to look in our organisms. So at that time, I was in a lab that was doing work on grasshoppers. Why grasshoppers? Because grasshoppers are great insects for this. They're big, and they got big cells, big juicy cells that you can easily work with. And you can do, these cool developmental interactions where you go in and remove cells and you inject cells so you can follow, all that kind of cool stuff. And we were also closely affiliated with another lab at Berkeley, Corey Goodman's lab, and they were doing Drosophila development. And Drosophila is nothing like grasshoppers. They're teeny tiny, they're really small. So you peel them open and you look in the embryos, and the embryos are even tinier. And you go into the embryos and the cells are minute. But of course, with Drosophila, you've got all these molecular tools. You can do all this genetics that you couldn't do with grasshoppers. What was really informative to me was, okay, here I am. I'm used to opening up a grasshopper embryo, and I look at the nervous system, and the nervous system looks like this cobblestone road. It's just a thin layer of cells at these early developmental stages. So you just see all these little cells there. First time I looked at them, I was totally lost. What am I supposed to see? It's just a bunch of bumps. But after a while, you get used to it and you start to recognize, oh, I know that bump. That bump is a cell called MP4. You got names for them. And it's in an interaction with these other cells, like these corner cells. And there's ACC and there's Q and all these cells that are talking to each other. And pretty soon you get so, you put on this thing that looks like a gray field of rocks as far as you're concerned. But the names of everybody. You can just spot them all. And then the first time I got to do this with Drosophila, I was, first of all, horrified at how tiny they were. But then you look at the nervous system, it's the same. You start spotting all the same cells scattered along the length of the nervous system. And you realize that these, and you know, and you start staining them and you watch and you can see all this growth, and the growth patterns are the same. You know, MP4, the one I mentioned, is going to form this longitudinal pathway, and Q is going to form this commissural pathway. And it's the same in Drosophila, which is astonishing because these animals are separated by almost 400 million years of evolution. So they're all identical. And actually at this time, the bread and butter of these two labs was that Corey Goodman's lab would isolate the molecules, doing a lot of monoclonal antibody work, and identify all these different molecules that are expressed in very specific patterns. And then they'd use the tools that you have in Drosophila to go back, figure out what the genes were, and you could start mutating the genes and pass that information back to the grasshopper lab. And we would act on that and say, oh, you just discovered this molecule is really important and it's expressed on this cell. Hey, we can go into that cell and we can pluck it out, or we can even go in and grab it and move it to a new location. And what happens with spurious expression of that particular gene in these places. So they're all related. Evo Devo is no big surprise. You know, there was, before Ernst Mayr and all these people had been arguing, no, you're not going to find any homology because 400 million years is so long and evolution is so powerful and natural selection is just going to shred that. You're not going to see any conservation. But right then we knew that there was all kinds of conservation going on. There were differences, subtle differences, but yeah, we could track those. Also at the same time, when I was at Utah, because I was known as a zebrafish person then, I made connections with the zebrafish labs there, and I had a collaboration with somebody who wrote to Nusslein, Volhard, and Wieschaus and said, hey, we're working on zebrafish. Can we take a look at what your genes are doing, your fly genes are doing in the fish? And so we also had this side project where we were doing all this analysis of gene expression in fish, where the genes we were looking at being expressed were all from flies. And there we got 700 million years of divergence in evolution. So yeah, it was kind of cool. So that's my answer to that question is, sure, we knew about these things. We knew about neutrality versus adaptationism. We knew about Evo Devo and deep homologies between different organisms. And, you know, when you're a scientist, you say, great, okay, let's go study this. I can make a hypothesis about this and I can go test it. So that's how it actually works. It isn't a big conflict. So, you know, nowadays there are some people, like Dennis Noble and some others, who are arguing for an extended evolutionary synthesis. And they're all posturing and talking about how we got to get rid of the old synthesis. We got to go to this new idea. And it's just self-serving nonsense. That's not how science works. We've got, if they've got legitimate good ideas, we will incorporate them into our work and they will just become part of evolutionary theory. You don't need to rename it. And no, I'm not going to give Dennis Noble credit for reinventing evolution. He isn't. He's just not. So that's kind of my answer to that question. That okay, yeah, there's stuff going on, but really, the questions being asked are kind of irrelevant by these people out on the fringe. Okay. Now I'm looking at the YouTube comments to see if there's any good questions there. Oh, somebody says they didn't get it. Oh, something was funky going on there. All right, we have questions, and I have a panel of people who will answer them for me, especially since we got some astronomers here. So one question on the YouTube chat is, can we offer you big answer to riddle universe planet Earth? We inventored great learning for open mind. Anyone? Yeah, I'm sorry. That makes no sense, but the astronomers can answer that, right?
We'll just say the
42.
answer. It's exactly what I was going to say. You beat me to it, Jody.
42.
Great minds think alike. The question is, what do you get when you multiply 6 times 9? 42 in base
Oh.
13.
Oh, okay. I was going to say, my biological mind is having some problems with that.
Well, that was in the book. The actual question he came up with when he was pulling the pieces out of the Scrabble bag is, what do you get when you multiply 6 times 9? So, I mean, Douglas Adams did this deliberately. I mean, to switch to base 13 and all that.
Yeah. Okay. That sneaky guy.
Exactly.
Yeah. Yeah. I wouldn't want to be a humorous novelist because they all seem to die too young.
That's for sure.
Okay. Let's see if I got a better question. I am told small scorpions have established themselves near the London harbor, hiding in stone walls without masonry where they fit in the tiny cracks. I would just ask, are they true scorpions? Because there are pseudoscorpions and whip scorpions, which are not really scorpions. I would be surprised if scorpions were thriving in a northern climate like that. Oh man, okay, here's one we can all work on. Would you please recommend books which are simple enough for non-experimental, non-scientists who would like to read about the various topics you discuss in your presentation? Okay, I'll turn it over to you guys first. Do you have basic elementary texts that you could recommend to somebody who's not deeply embedded in your field?
As in general, just general audience books.
Yeah.
There's a book that just came out not too long ago by Max Tegmark called I think it's called The Mathematical Universe. And that kind of takes you through some, like astronomy from Big Bang to the cosmic microwave background into the funny, crazy area of the multiverse and quantum mechanics. And it's kind of an interesting read. It's his journey along with general science for all intents and purposes. If you're into that kind of stuff, I would recommend that book.
Okay. Yeah. Astronomy actually has a little bit of an advantage because you've got that Carl Sagan tradition. So you can always name.
I was going to say Cosmos, but I thought that would have been the obvious answer, perhaps.
Yes. Yeah, and I recommend Carl Sagan all the time to my students, except not Dragons of Eden, which is terrible. He should have go ahead.
I was going to say Cosmos as well, because you can also go to this TV series, right, and watch it, and then read the book, or read the book and watch it. So you get the best of both.
Yeah.
Yeah. And Cosmos doesn't just go over astronomy. It talks about science in general, really. I mean, if you really read it.
Right. Yeah. I also tell them to go watch Jacob Bronowski and The Ascent of Man, which is another good series that covers a lot of ground. It's not just physics. He's a philosopher, so he knows everything apparently. But that's another good one to recommend. Also, there's Sean Carroll. A lot of Sean Carroll's books are excellent, actually. Yeah.
He has really good podcasts too, that he has about just bunches of topics that he talks to different people about. So, and Sean B. Carroll, there's two Sean Carrolls. He also has a good book that I read, the Serengeti Rules, I think it's called.
Oh, right. Yes, he's got a whole bunch of good books.
Yes.
We actually use some of his books in our introductory biology course because he
Oh.
gives such a good introduction to evolutionary theory and things like that. So yeah, he's just go look up Sean B. Carroll. And
Uh-huh.
Who's the young girl that put out a book recently, or was it a YouTube channel? I don't know. She's like 12 or something, and she wrote an amazing book, but trying to remember the topic. I thought it was maybe evolution or biology or something. I'll figure it out.
Oh, well, that
She's
sounds interesting.
Yeah, I mean, and it's a really, really good book because it's, it's not creationist or anything like that, but it's geared towards middle school kids.
Yeah. Yeah, I've also been reading, let me see, do I have it? I have it right here. Yeah, I got this book. So,
Okay.
yeah, so Diary of a Young Naturalist by McAnulty. It's written, it's apparently a condensation of a blog, which is kind of nice. But this is a guy who's just a young man who is just crazy about nature and is making observations. So he's Irish, so of course it's beautifully written, right? I don't get it, but they've somehow got that gene. No, they don't believe in genes like that. Anyway, so this is what I've been reading as good exposure. Books regarding my research, the questioner qualifies. Yeah, that's tricky. That I feel like there haven't been any great books written in my field. There's been some good ones. Authors I recommend are people like Wallace Arthur, who has written a number of books on development and paleontology and things like that. I disagree with him on some things, but in general, he's a pretty good source. Yanni Nusslein-Volhard wrote a book. It's a very German book. It's an overview of developmental biology concepts. And it's a little bit dry, but it's written for a general audience, and it's pretty good for getting the foundations for things like, what is induction to a developmental biologist? Developmental biology appropriated a whole bunch of words from psychology back in the 1920s and 30s, and it confuses people so much. For spiders, let's see, what would I recommend? Oh, they're all behind me, behind my green screen, so they're a little hard for me to get at. There's a bunch, there's Spiders of the World. Let me look it up. I'll get the specific title for you. There have been several really good books. And
If I may, for evolutionary biology, I've always enjoyed Carl Zimmer's books.
oh, Carl Zimmer's terrific. Yeah.
They read like fiction,
I know.
In that they are easy to read and you just fly through them.
Right. And he's also correct on so many things.
Well, that's nice.
Yeah, one of the things that just really impressed the heck out of me is, his book, She Has Her Mother's Laugh. Whatever. Yeah, that's, I kind of saw that title and was terrified that it's going to be one of these genetic determinism books that are so popular among the alt-right and so forth. And no, it's not. He's actually very careful about pointing out the boundaries between what you can know about genetics and what's epigenetic and what's cultural. And that's the theme of the whole book. So it's really good that way. Another book I'll recommend it just came out just recently. Was it last summer? Spiders of the World, A Natural History by Norman Platnick. Platnick, I unfortunately never met him, but he has this amazing reputation in the arachnid world. Platnick was a Smithsonian spider expert, so an arachnologist at the Smithsonian, and he did everything. He just published so many papers and categorized everything. Spiders of the World, in some ways, was kind of a disappointment to me because I'm usually looking for detailed books to help me figure out what the heck that spider over there is. And it's not that. It's like it says, it's a natural history. So it's this beautifully illustrated book of all these different groups of spiders. It goes in at the family level, not the species level, which is great. So because there's so many spiders, you go in at the family level and you can get a good overview of them. And he talks about behavior, he talks about silk production, talks about venoms. It's just a really nice book for getting an introduction to spiders. It won't help you identify the spiders in your house. For that, I use this book a lot. Spiders of the North Woods. It's specific for northern United States and Canada. So all the people here, all three of you, you could use this book. It's just a slim little guide to spiders that occur up in this part of the world. So, and it's really easy to use. It's short. It's kind of nice that way. Lots of pictures. Yes, I feel like such a big boy because I can say my favorite books have lots and lots of pictures. And that's true of this one too. So Spiders of the North Woods by Larry Weber, a really useful guide. If you're farther south, you just go look for your geographic region and spiders on Amazon. A couple of years ago, before back when we had real in-person meetings, the American Arachnology Society had a meeting, and they were just handing out these little pamphlet style guidebooks to, for instance, spiders of Kansas. It's really handy because when you're out in the field or when you're in the bathroom, you don't want a big bulky book. You want something like this or even smaller to do. I've actually been contemplating doing a little book for my students where I would, spiders, the University of Minnesota campus, which would be fun to do. Tell them here's where you can find grass spiders. Here's where you can find some amazing Parasteatoda. Here's where the wolf spiders are hanging out. Just wake up and look at, one day I'm walking around on the campus and what do I see? I see a jumping spider ballooning. It's just kind of floating down to me. So, if you're awake, you can see these things. So get your nose out of your cell phone and look around you, and there's spiders everywhere. So those are some of my recommendations right there. Okay, let's see, where did I lost my connection to, I'm sure it's on here somewhere. Oh, there we are. Yeah, so books regarding my research. My research has not been published. It's all preliminary stuff where I'm just trying to figure out how to do this sort of thing. And most of the work I look at is just published papers. It's that esoteric. So yeah, what can I say? Get on PubMed, just search for spiders. Look for anything in the last 10 years. They're having a little bit of a renaissance in my interests in that there's a huge amount of spider literature that is just systematic. It's just going around figuring out who's who and what's what. But there's currently a lot of stuff being done on developmental biology of spiders, which is kind of cool. I got a paper a while back. I think, Berger, did you send this paper to me? This cool paper about opiliones, daddy longlegs, and they're doing experiments on the genes in daddy longlegs, and they're making stubby-legged daddy longlegs by identifying the genes involved in lengthening of the limbs and doing knockout experiments on those. Okay, we are coming up on an hour, and I don't want to bore everyone, so I will generally stop the stream at about an hour, coming up in 2 minutes. So this is your last chance. Anyone want to make any comments? Go ahead. And any of you beautiful people who have joined in, if there's any last words you want to say. Yeah, make me do all the talking. This happens.
Yeah.
This.
That's all, folks.
Yeah. Go ahead.
How often are we going to do this? Every Sunday?
Oh, not every Sunday. I do it like every 2 weeks, every month. We'll see.
Oh, okay. Sure. Whatever works.
Yeah. I'll make an announcement on the Patreon page every time.
I'll be very curious to find out what happens when you go back to class.
Oh, me too. And I hope it's not this.
Yeah.
No, I was just going to say, I appreciate your blog. I've been reading your blog since before Free Thought blogs.
Oh, back in the science blogs days. Yeah.
Yeah, so
been around a long time.
I have to say I don't read every single thing
You
you write.
shouldn't. Yeah, I don't blame
I
you.
cannot. I don't know how you do that, but I do appreciate a lot of the stuff that you write about education and some of the other things that I said I didn't want to get political about. But I do. I appreciate your blog. So thank you.
Thank you. Yeah. Yeah. So I'm sure I will be reporting more about the classroom experience in the coming couple weeks. I'm dreading them a little bit, but maybe it'll be better than I fear. We'll see. I'm also planning to do some stuff. Like I said, I'm going to my son's promotion ceremony back on the West Coast. And we are not going to hang out on an army base for 5 days. That would be boring. But we are planning to go hit up the Pacific Coast and do some tide pooling. My granddaughter's coming along. So we've got to teach her the wonders of going out to the ocean and finding weird critters all over the place. So we're going to be doing that. So I'll probably be reporting back on that. Maybe I'll make some videos of a little girl getting stunned by all the things she can find in the ocean.
That's in San Diego, you said?
No, Washington State.
Oh, Washington State.
Yeah. My favorite place on the planet is the Pacific Coast, the Quileute Reservation up along the Pacific coast of the Olympic Peninsula. Just gorgeous stuff and relatively untainted and untouched. Not completely untouched, nothing is anymore, but there's some great stuff to see there. So my whole family is going out there because I took them there multiple times when they were growing up and always raved about it. So we've started a tradition. So that's my plans. So maybe we'll hear back about that. And people should keep sending me questions, and I'll just stack them up and we'll talk about them in a couple weeks. Yeah, I should probably plan on just doing a report on the state of education at the University of Minnesota Morris in a couple weeks. You guys can report back about state of education at your places. We'll talk to you all later. So I will end the stream right here. Thanks everyone for showing up.
Thank you.
Yeah, thank you.
Thanks.